Okay, welcome everybody to the Pygor tutorial. If you would like to follow interactively, and we hope you can, please now either make sure that the first command runs in your Jupyter notebook. Carlos will go through how to start the Jupyter notebook, but the first command is this. I'm putting it into the chat window. If it doesn't run, you can fix it later. For now, we recommend using the collaboration notebook, which can be downloaded here, and run the first cell now, since it takes a few minutes to run. So while I talk, you can run it. So I will repaste that in a few seconds for those that are just joining now. In the meantime, so please run that first cell in the collab notebook right now. So Pygor is a software for analyzing lymphocyte repertoires. It is linked and based on the IGoR software that evaluates the probability of generating a given TCR or a given BCR from sequencing data. Pygor was developed by Carlos Olivares Rodriguez, Thierry Mora, and myself, thanks to an ERC proof of concept grant, and builds on the work of many other people, including Anand Murugan, Kantan Marku, Yuval El-Hanati, and Kurt Callan, among others. So thank you to everybody who contributed. Pygor is a Python interface that calculates statistics and repertoire properties. In principle, you don't have to be a super great programmer. It's based on Python. And today we will focus on some applications, but mostly applications and commands. So if you have comments about or questions about the commands, applications, or how to use it, please post them in the chat or ask them directly. And we will address them as we go along. However, if you have problems with installation or running the commands or commands not working, please send us an email or post it in the chat or post it in the Pygor help web page on GitHub, and we will answer these, but maybe not during the tutorial. We'll get back to you about them. Okay, so with that, thank you everybody, and I will hand it over to Carlos.
Okay, thanks, Alex. Good morning, everyone, or good afternoon, wherever you are. So as Alex mentioned, there is a collab version for this tutorial that you can access from the website. This is the link. I guess someone can put it in the chat if you want. I'm gonna put it in case there is someone that couldn't install Pygor in their system. So the first thing that you need to do is just run this cell, as Alex mentioned, and it's going to be like a warning, and just accept that. And this is just a virtual machine that is going to run, it's going to install everything that you need to go for the Pygor tutorial. But for the rest, the people that could make the installation, so now you can just run Jupyter Lab. And by the way, so if you have some installation problems, some issues with Pygor or IGoR for this tutorial, we can check that in the issues here. I'm gonna pass this also in the chat in case you have some other questions about it. Run the Jupyter Lab. So in this Jupyter, please open the tutorial IPython notebook, which is the one that is showing here. Right. So so as Alex mentioned, Pygor is a Python interface for IGoR, and IGoR is a software that allows us to evaluate and generate adaptive immune receptor sequence using a BDJ recombination model, a probabilistic recombination model. So, and kind of the focus of this tutorial is to show you how can you evaluate sequences and generate synthetic sequences, and also infer new models given your own data and your own genomic templates and your anchors for for these templates. So as probably everybody here knows, the VDJ recombination is a stochastic process that assembling sequences using taking genes, specific genes, to combine it and generate a specific sequence. So one sequence can be constructed from sorry, one scenario determines one sequence, but one sequence can be constructed from different scenarios, right? So I can select a different B, a different J, or different D, and the number of insertions in between will change, and the deletions, but the sequence will be the same. So all these scenarios, IGoR is gonna find these possible scenarios and assign a likelihood for that. And this is a process that is an optimization process. We're gonna run iteratively inside IGoR. And this will define the parameters of the model, which are the probabilities that it's gonna find. So all this information about the model is what defines a model for IGoR. So IGoR already has some default models for humans and for some mouse. And for this tutorial you're gonna use a human and a T-cell receptor, a beta change of a T-cell receptor. So please run the first command using to load first the Pygor package. And to do it is just run import Pygor3 as P3. And from now on, all the commands for Pygor is gonna be referred from Py3 in the same way that you probably use NumPy as NP. So let's just run the first lines. So what you have here is you get a default model from a default model for the human and beta T-cell receptor, and all the information of the model. That means that these probabilistic parameters are encapsulated in this variable. So this variable called MDLHD is just an IGoR model object. So to see whether the parameters of this model, you can just export to a PDF file. For instance, this is gonna show you some plots of the conditional probabilities. You can write wherever the name you prefer here, and just export the plot models. And if you execute this, you're gonna generate. You're gonna see all the images of the model. So as you can see here, first it shows you the structure of the bias network for this model, in which, for instance, you see that the J gene depends on the B choice, right? But the B choice depends on nobody. So if you see the probability of B, of choosing of the B choice, is gonna be this one, where you can see what are the alleles that are much more probable than others. And also the other conditional probabilities, as I mentioned before, in the bias network I show you, the J gene depends on the B. So you can see here, for instance, for this gene, you have this value of this probability, and again, you have all the events on the model plotted with this just one command that you can visualize and see and extract your own conclusions from that, right? So, so if you want to access these values, you just need to specify the name of the event. For this case, is gonna be J choice. So this is gonna give you the probability of choosing one J, given that you selected one P gene specifically. So when you execute this command, you can see the probabilities here of these conditional probabilities. You can see the IDs of the event, of the realization of this event, and also the names, for instance, or the sequence that are part of the genomic template, right? So again, if you want to see a specific value of what is the probability of getting this J3, given that I choose the one with the ID 7, you just run this command and you will have this value, right? So all of this defines the parameters of the model. And if you want to plot a specific event, you can do it. And just in this example, I'm just doing the J choice. So just use the command plot event, and that will show you the specific conditional probability for that. So that's something that is coming by default by IGoR, and also these are conditional probabilities, but for some reason you may want to see the marginal probabilities of the event. So to get the marginal probabilities of the event, you just need to sum over the variables that you want to sum out. So Pygor does do that automatically for each model. So you can just export again in the same way that you did for the conditional probabilities. You can do it for the marginals. And again, you just select a name to save the PDF file that will be generated. Sorry, there's a question?
That was somebody who didn't mute themselves. Carry on.
So you have the marginals, and again, it shows you the structure of the network, but this time there are no conditional probabilities anymore. There's only marginals. So this is the probability of selecting one specific J choice without by chance. So and again, for the rest of the events, for the insertions and the deletions, and as before, if you want to get the values of this marginal, you just use this variable p marginal, and you will get again the marginal probabilities and also the information about the genes.
So what we're looking at here, Carlos, are the marginal probabilities of the model, the things you plotted?
No, okay, what I'm showing here are these marginal probabilities that I just plotted. And let me to emphasize that IGoR has these files that has marginal files, but these are not the mathematical marginals, in the sense that you marginalize over all the variables. These are kind of these are the real marginals for one variable. Right?
So Pygor has the advantage of actually giving you proper marginals in the sense of summing over all the other variables that enter the probability.
Yes, exactly. Yes, you get these marginals as a calculation from that Pygor does. He used a variable elimination algorithm to do it, because it's a little bit tricky for some of the events, so you can do it manually, but you already have it. And again, if you want to plot this event, you just use the name of the event that you want to marginalize, and you see specific plots that I showed you before. And also, if you want to have some joint probability.
Sorry, Carlos, we have a question here. From which data set are the human TCR probabilities inferred in this tutorial, the model baseline?
From the Emerson data that was I can. This is specified in the IGoR paper. That you can
Okay,
check.
I think
later.
if you said Emerson, everybody who knows about data sets understands what that means. So that's good. Thanks.
Okay. Okay. So okay. So as Pygor also, you can calculate joint probabilities with Pygor. This is something that Pygor does in the same way that it does for it do it for the marginals. So you just need to specify which events you want to get the joint probability, and use this command get p joint, and you will get the joint probability of this. So, and of course you can plot it and get a graphical representation of that. You must notice that of course this is different than the conditional probability, because this is the joint probability. This is the probability to get 1B and 1J by chance at the same time, without no conditional, right? So other things that Pygor that you can have using Pygor is to calculate the entropy of the model. So
sorry, maybe before we go there, so basically these commands with plot used in the same way as a normal Python plotting tool on NumPy basically allow for visualization for all the marginals and all the distributions you may be interested in the model, right?
Yes. Yes.
Okay, so
okay. So so to calculate the entropy of the event, you can use this command plot recombination entropy, and this will show you how the entropy of all this model, and this entropy is it gives you a sense of diversity in your model, right? So what it does also can show you the part that the diversity given by the the selection of the genes, by the insertions and then deletions. And as you can see here, you have the one that contribute much more for the diversity are the insertions in between the B and D genes and the D and the J genes, right? So, and if you want to get these values, you just need to apply this function that gets data frame entropy decomposition, and you will get all all these values here in a data frame. So and also you can get the mutual information given this model. So you just need to request the model get the mutual information, and it will give you a data array. And you use the command plot mutual information for that. And you will see here, for instance, that okay, the D5, the deletions in the 5' side are pretty correlated with the deletions in the other side. But this is not so correlated with the B choice or the D or other events that you can see in this plot. So this gives you a sense of correlations between the events, right? But all of this is what configures a model, an IGoR model in particular. And we're going to use this model to evaluate sequences. So what it means, evaluate sequences? Evaluate sequences is going to is like get an annotation of what are the scenarios that make this sequence that can make this sequence possible. So to do that, we'll just start with one sequence. So
right, so maybe to recap a little so far, what we've been doing is we've just been talking about the elements of the model, of the ready-made model that ships with IGoR built on the Emerson database, and gives showing you how you can examine the elements of the model. If you build a different model, these elements will look differently, but you can go in one by one to any level of detail and examine these elements. Now, if you have new sequences of your own that you want to evaluate with this model or another model you build, how do you do that?
Yes, exactly. That's what we're gonna do. So for any means, if you already have an IGoR model, you can use this model to get to evaluate the sequence, right? So so we have this sequence. This is a test sequence, and we're gonna pass this sequence to the Pygor evaluation method, and you need to specify the sequence, and this variable could be a string, as I put it here, could be the NumPy array, a list, or a Panda series or data frame. And then, of course, you need to pass the model, and you need to specify the number of scenarios, and these other variables are just if you want to keep the information in your local disk or not, right? So let's run this command, and this is gonna run IGoR in background. So once it's gonna, you see the commands that is used here to run IGoR, and as you can see here, I pass these values to this variable, and with this variable you could see how this sequence could be constructed. So the sequence could be constructed by the 48-bit choice and that AGJ gene, and this is the insertions IDs, and it gives you the probability that this is the scenario given that you provide that sequence, right? And also it gives you a PGen estimate. That means that what is the probability to generate by chance this sequence, right? So as you see, it's a very low value, 10 to the 16 for this case. So this is kind of the usage that you want you're gonna make when you have your sequence. I want to see how this sequence has been constructed. And to visualize a scenario, you can select, for instance, the first element of this row, and to just make a model, please plot the scenario, and you pass the scenario, and you will get some plot of the scenario.
So right now you're plotting the most likely scenario, is that
yes,
it?
Yes, exactly. This is the one on top of that, here on top, the one that has a 0.17 of probability, this one. So, and these blue lines, they are mark the position of the anchors. So the anchor is the anchors of the germ lines are the conserved cysteine for the B gene, and the conserved phelanine and tryptophan for the J gene allele. So this in the model, you can specify the anchors, and you will get this plot with the marks of the CDR3. So, and if you want to plot all the scenarios, you just make a 4 over all these scenarios, and you will have all the different scenarios in a visual way. And as you can see here, the one that which is much more probable is different for the second most probable. For instance, here we have a bunch of insertions. Yes?
All of these scenarios have the same PGen.
Yes, all these scenarios have the same PGen, exactly, because it's the same sequence.
No. So
no.
The answer is no. They don't have the same PGen, but the PGen that's given is for the sequence, not for the scenario.
All of them together make PGen.
Okay,
No.
okay.
You add all the probabilities to get PGen.
I got you.
That's a bit confusing. The PGen.
Oh, okay. The relative probability. Okay, it's the third column.
Yes,
scenario
exactly. This is a relative probability, given that you have the sequence.
Thank you. Thank you.
Okay. And the pigeon is the value to generate the sequence.
Right.
So if you want the probability of the pgen of scenarios, you need to multiply the pgen of the sequence times the relative probability of each scenario.
Got it.
Yes.
But this is maybe probably too intricate for the average user. Basically, we're looking at one sequence and we're looking at all the scenarios that are likely to produce it and examining them by one by one, and they all have different probabilities of generating this sequence.
Yes.
Mm-hmm.
Yes, exactly. Well, so these are the evaluation part. So with this, you can see how the scenarios have been constructed, and you know the probabilities for that. And if you want to see whether the parameters, the genomic template for that, you can just run genomic data frame. And in this case, I showing the J gene. And as you can see here, I have the anchors, the sequence, and the genome, the gene notation and the gene description. Right. So, but now, so this is just for evaluating the sequence. Now, because we have this model, we can generate synthetic data. That means that we can use this recombination process to just draw some sequences. So this should be easy. Just use the Pygor function generate and pass the number of sequences you want, and again the model, and this will give you the sequences that you request, sample from this model, right? So, and as I mentioned before, one scenario defines a sequence, but a sequence could be defined from many scenarios, right? But because we generate this sequence, we know exactly what is the scenario that constructs this sequence. So because IGoR select that scenario. So you just, if you want to see the values of that, just put return scenarios true, and you will see all the realizations for these sequences that you just generate.
Carlos, we have a question. Could you generate
Yes.
sequences which look like another sequence, or are some distance in space to a given sequence?
From some distance in space to a given sequence.
Yeah, I think the short answer is no in the
yes, I think so. I mean,
the way you would go about this is that you would generate a large number of sequences, and then you would have to select those that are close enough depending on your criterion. So it's a bit brute force, but in the current version, that's the way to do it.
Yeah.
But if you think that's an interesting development, a student of ours has done it, what you're asking for. We haven't really included it in software so far. But yeah.
Yes. Okay.
Thanks for the question. Good.
And just to just so okay, so from this you can get the sequence, right? And you use this sequence in the way you want to use it. So, but all of this has been, if you already have a priori a model, an IGoR model. But if you want to construct a new IGoR model with your own genomic template, with your own anchors, or the ones that are in the EMGT database, for instance, or whatever your own species that you want. So for your own data, you can do it. You can construct IGoR models in a very simple way with Pygor. So to do that, first we're gonna load these samples of sequences, and again we only care about the nucleotide sequences for this case. right? So you only care about the sequences. And for this particular,
so these are the sequences on which you're trying
sequences,
to learn a new model, right?
yes. So these are the sequences that I just get from a sequencing machine, for instance, right? So we know these sequences are human TCRB. So we're gonna infer a new model with the genomic templates that we can download automatically from EMGT using Pygor functions for EMGT. So if you want to see what are the available species, for instance, from the EMGT, just run Py3 EMGT, get the species list, and you can check or you construct your models in principle for all of this.
So this is also an sort of an addition to Pygor that IGoR didn't have, that it in an automated fashion can find genomic templates in databases, correct?
Yes, exactly. Yes, for now it's just for EMGT, which is probably the most popular. But yeah, you can download any species you want from here, and you need to specify score the species and the change receptor change. So for this case, because we know this is our human TRB, we're going to select the Homo sapiens here. And then you will say to Pygor, the EMGT functions for Pygor say download this reference genome for this species. And we do that, and it will automatically get the information from the EMGT. And if you want to see in the EMGT website, just click on this, and you will see where to get these genomic templates. Oops. Right. So now we have our BDJ sequences. Right. So we have the in the pandas data frame the genome, the germline sequence, the name and the anchors, and also the functionality. So with all this information, with this dictionary of data frames with the genomic templates. So for instance, if you have a new a human or a mice or whatever that you generate or you engineer your own genomic template, you can add it here, and then you can construct your sequence just by make a default model from this data frame. And this will generate a model with a uniform distribution for all the probability distributions in the model, for all the parameters of the model, right? So because this could take a long time, we are gonna just sample a small part of the sequence and pass it here, right? And usually, if you use another software to know if your sequence is productive or not, IGoR needs to know if your sequence are productive or not, because to infer a model. Okay, so what is a productive sequence? Basically, a non-productive sequence is a sequence that has some stop codon, or is not multiple of 3, or basically that. So the IGoR can use this model or this genomic template to make a simple alignment and determine if the sequence is productive or not. But if you have your own data that you know that which ones are productive and which ones are not, you can just pass this step and go directly to the inference process. So because we don't know a priori for this sequence, we are gonna run this command, and this naive alignment is gonna give me, as I said before, the functionality and also the CDR tree of the sequence, right? So once it finishes, I'm gonna show it here. You will see that, for instance, for the sequence 0 is not functional, but the other one it is, and so on and so forth. And it tells you because it has a stop codon or not. And it gives you all the information. And again, you get the CDR3 sequence because that CDR3 is a deterministic part of the sequence. So, and again, because IGoR needs the non-productive sequence, because the productive sequence, the ones that are has passed some selection process, and this selection process could put a bias to infer a recombination process. So the ones that didn't pass the selection process are the ones that has never expressed. So these are the non-productive sequences. So we are selecting these, the ones with the functional, that are not functional from this small sample of datasets, and we have the non-productive sequence, and we are going to use this non-productive sequence to infer a new model. And to infer a new model, you just need to do like Pygor3 infer, pass these sequences in, as you see in a Panda series, and pass the initial model that we create with the uniform distribution. And for this case, we are selecting the 10 iterations. And to see the convergence of that, we are returning the likelihoods. So it's gonna align this sequence, it's gonna start a process of iteration. So because we pass the 10 iterations, it's gonna make these 10 iterations. And after that, you can see the mean likelihood, mean log likelihood, and check how is the convergence to the final parameters that you have for your that you just get from your model. And once you infer this new model, you want to see, okay, what are the characteristics of the model, and to do that, you just need to run the first command we run before, which is export conditional probabilities, and you will see again the Bayes network, the probabilities for B, the probabilities for J given B. And because this is a very small subsample, just to show you here in this tutorial, of course we have a lot of zeros because we didn't cover enough of the space to get a good model, in a sense, a good predictive model. And so that's it for now. If you have any questions, so please go ahead. Pass to.
So thank you, Carlos. Yeah, does anybody have any questions? Hopefully we managed to solve most of the error message issues as time went on. Anybody, any other questions? So the other, we tried to concentrate today on some specific applications. IGoR also makes it easier to interface with Olga and Sonia, which we don't really have time to give examples for today because we wanted this to be sort of self-contained. There's a question from Dan Hudson: for future reference, some commentary markdown on the tutorial would be helpful so we can work out what each step is trying to achieve. Right, so we can put that up. There's also other notebooks in the tutorial folder that go into more details. If you're interested, then, and have examples of other commands. They're here, the D01, D02. They also have some of the same commands we covered today, but then go into more details of each one of them, which we didn't want to do online. but they're sort of organized roughly by number by the themes we covered today. So you can have a look at those. We will try to also, of course, comment them in a better way and comment this one. Thanks for the suggestion. Anybody else?
And just to respond to Nicolas Perrin Gilbert, so it probably using an old version of IGoR, because right now there is a new version of IGoR, well, IGoR 1.2. So here is you probably need to update to the last release for that. I don't know if someone else has other questions or problems during the execution of the notebook.
So maybe if there are no other questions, maybe what we can do is I'll stop the recording here. Thank you, Carlos, again for the tutorial and the presentation.
Thank you all for